Very good morning. So in this video lessons, we are going to discuss about uh, varietal seed production in rice. Actually, rice that have both variety and hybrid. First, we can discuss about that matter related to that varietal seed production. So before getting into that production related data, I wanted to give you some information about that seed structures. So that rice seed, that is a monocot, that will have only one cotyledon. That cotyledon also fully utilized by the endosperm during that seed development process. So that resulted in reduction of the size of the cotyledon, that is available in the seed in the name of that scutellum, that also placed in between embryo and endosperm. Then it's albuminous kind of seeds, and the seed is surrounded by the lemma and palea. So here you can see that the lengthy covering is a lemma, this one is a palea. So that lemma and palea that completely cover that endosperm as well as that embryo. So the major area of the paddy seed is occupied by the endosperm, and very small part it's allotted to that embryo. So that is, this is embryo. The embryo is a living part, whereas that endosperm it's a non-living in nature. This is in short about that paddy seed structure. And then regarding that rice plant morphology, so here that flower is called as a panicle. So the flower is the panicle, so that it will have topmost leaf is a flag leaf, and it's a fibrous root systems. So monocot is a monocot plant. This don't have a well developed stem and all. So it's a stem and all covered by the leaf. It's not a woody in nature. So these are all the things with reference to that rice plant morphology is concerned. Then before starting that any seed production operation in any crop, we must know that floral biology of that particular crop. Particularly, we must know that that pollination behavior and the time of opening of the flower and the flowering period. So if you know all these parameters, so based on that you can select a land and you can plan that appropriate timely intervention related agricultural operations. So most of the operation we have to do it in a timely, then only we can get a very good yield with assured seed quality. So for this purpose you must know that flowering behavior of your seed crop. So here that the seed crop is a paddy. So the paddy is a self-pollinated one. So the extent of cross pollination is 0 to 4%, but this is a self-pollinated one. And that flowering time, that anthesis, that is the first opening of the flower. So normally that flower opening will occur in between 7 a.m. to 12 p.m. That flower opening also is a basipetal nature. Basipetal means that in the first the flower is a panicle. So the topmost flower first to open, then that proceed towards downward. So that is a basipetal nature. The another type is there, that is that we will come. I will come to that one and the remaining crops while we are discussing that maize and kambu and all. Whereas in the case of rice, the flower opening is a basipetal. The inflorescence is called as a panicle. Normally that panicle will be ready for pollination fourth day after emergence from the flag leaf. So the panicle will be ready for pollination 4 days after panicle emergence from the topmost leaf, that is a flag leaf. And flowering period also that will be for about 8 to 10 days. Some of the female parental line, so normally we will use that A line as a female parental line for hybrid seed production. So some of the female parental line, the flowering period is 10 to 12 days. So in this case that will take 12 days for complete flowering of all the plants available in this particular field. So in general that paddy, the flowering period is 8 to 10 days. So these images related to that flower, so cell pollinations. Then here you can see it's a basipetal thing and acropetal and basipetal. So in the case of basic petal, the topmost one is open first, and then that proceed towards the downward. Whereas acropetal, that the bottommost flowers will open first, then proceed towards the upward. So that is a process. So in the case of a paddy, it's a basic petal nature. So here you can see that topmost flowers available in the panicle is started opening and slowly towards proceed downwards, and finally the bottommost flowers will open. So this is a process with reference to that flower opening. Whereas, so this is a internal structure of the flowers. So this is a stamens. So this is a stigma, two stigma, pipette nature, and this is a ovary. Green color is a ovary. So here you can see, so that ovary, everything is available. So this is surrounded by yellow color, that stamen. On either side, three things will be there. So totally 6 anthers will be available. So if it is a fertile nature, so the appearance will be like this. So if it is a sterile in nature, that stigma and everything will be that won't be any profused growth, and that pollen grains also somewhat papery in nature, not in lemon yellow color, so that will appear somewhat brown or black in color. So these are all the internal floral morphology of that paddy. And also we have some pollen viability testing also. With the help of that also we can verify the viability of the pollens. So now we are getting into that paddy varietal seed production aspects. So now we know that pollination behavior of the crop. So the paddy pollination behavior is self-pollinated crops, and in Tamil Nadu more demand is there for paddy varietal seeds also. So based on that these two criteria, that is whatever the crop in which that self-pollination occurred and heavy demand is there for seed, and low SMR is recorded, means in these cases we can go for that 5 generation system of seed multiplication. Whereas in the case of paddy, SMR is not that much low, here 1:80, but more demand is there for seed. Then this also a self-pollinated crop. So because of that, in the case of paddy, we can follow that 4 generation system of seed multiplication, that is a breeder seed, foundation 1, foundation 2, then certified seed. The certified seeds will be distributed for the grain production purpose. So now we decided that number of generations for multiplication of that variety of the paddy. So after knowing the generations, so what are maybe the class? So these are all the things, agronomic things we have to select. So first one is the land selection. So regarding that land selection, already we have a thumb rule that whatever the land selected for cultivation of that seed crop in this particular season should not be raised with the other variety of the same crop in the previous season. Suppose the same field, the selected field is raised with the other variety of the same crop in the previous season, means there we have to follow some strategy to mainly to eradicate the previous season seed material, if at all anything is available in the field. By that way only we can overcome the problem created by the volunteer plants. Okay, the previous season crop, that seed may be remain within the soil. That seed will also germinate in the current season. So that is the volunteer plant, that is a pollen contaminant. So that will create problem. Because of that only that the seed certification department, they are insisting us that the selected land should not be cultivated with other variety of the same crop in the previous seasons. So in order to overcome this particular problem, what we have to do is you go for flowing of that selected land, then irrigate the land, allow the seeds, whatever the seeds are available in the field, allow the seeds to germinate. Once that seeds and all germinated, then you can go for that plowing. By that way you can kill all the plants. So by that way you can avoid the occurrence of the volunteer plants. So these things we have to follow. Apart from these conditions, the selected land should be a fertile one. Okay, so the availability of nutrients, so that nutrient availability in the soil, that also play a very crucial role with reference to that pollen development and pollination, proper pollination, and further grain development, all those things, further seed development, everything is decided by the nutrients that are available in the soil as well as applied nutrients also. Suppose the soil is have the problem of that salinity and sodicity, so there that will have more amount of that sodium and sodium related salt will be there, so that normally causes problem with reference to seed germinations. So that seed germination will be the problem. Once that excess amount of the sodium enter into the plant system means that plant tip burning, yellowing, all those problems will come. So the plant is not able to perform very well under that particular conditions. If at all that selected land have the problem of the iron deficiency, that pollen production potential of that plant will be decreased. The quantum of the pollen produced will be decreased. Once that quantity of produced pollen is decreased, there is a starvation with reference to availability of pollen for pollination fertilization of the female organs available in that particular plant. So ultimately end up with low fertility and low seed yield. So these are all the problem. So in order to overcome all these problems, the selected land should be a fertile one, and that should be free from volunteer plants, and that should have a good sunlight and proper aeration must be there. So sunlight availability also we have to ensure. If at all any trees are going to be there in and around your seed production field, so whatever that crop that directly that is placed nearer to the tree, because of that shade, that sunlight reception will be low, that yield that performance of that particular crop is not that much good. So proper sunlight and aeration also very important one. And I already told you we have to avoid alkali and acidic soils. Better we can have a clay loam with pH of 6.5. That is an ideal soil for cultivation of that paddy. So this is with reference to land selection. So this all comes under that agronomic principles. And then season. So season and climate also very important. That is a deciding factor. So as far as Tamil Nadu is concerned, we have so many number of seasons. So many number of seasons are available. So in this particular table, that different seasons and that coupled with that month. Say example, December January means that is a Navare season. So during that season, we can cultivate that rice variety with a duration of 120 days, so less than 120 days durations. Similarly, Swarnavare, Kar and Kuruvai, in all the seasons we can cultivate only that short duration varieties. And in the month of July, August, that is a early samba season, then August samba season, and late samba and late pisanam. So this and all, water availability will be more, and because of that, in samba as well as that late sanam or late taladi, during that time we can cultivate that variety with a longer duration, that is more than 130 days durations. Whereas during Navare, Swarnavare, Kar, Kuruvai, during that time, that sunlight intensity will be more and water availability also a limiting factor. So because of that, during that time, better we can cultivate that variety with a shorter duration of less than 120 days. So short duration variety we can cultivate during that season. So this is a season with reference to Tamil Nadu for cultivation of that rice is concerned. Then now that land also selected and the season also finalized, then we have to look for that seed material. So we have to use authenticated seed material, and appropriate quantity also we have to use. Optimum seed quantity, seed rate we have to use. Then only we can maintain adequate population in the field. So we have to maintain optimum population in the field. That also very important. That also insisted by the seed certification department. So I already told you, in the case of rice we have a short duration variety, medium duration and long duration. So normally short duration means that is 105 to 110 days or 115 days. Medium duration, that is 110 to 120 days. Long duration means that is 135 days or beyond that. So if it is a short duration variety means in that case we have to use 50 kg per hectare. Medium duration variety 40 kg per hectare. Long duration variety 30 kg per hectare. So in the case of short duration, and that spacing, whatever that spacing we are going to follow for at the time of transplanting, that is low when compared to long durations. So in the case of short duration, we will accommodate more number of plants. So because of that, there we have to use more quantity of the seeds when compared to long duration and medium durations. And then after selection of that seeds would be given with appropriate treatment. some of the paddy variety that have the problem of the dormancy. Not in all the cases, very few cases that have the dormancy. It's a kind of a physical dormancy. So in order to overcome that particular dormancy, the seed must be given with potassium nitrate treatment. That is, here we have to use 0.5% potassium nitrate solutions, and the seeds should be soaked in the potassium nitrate solution for about 16 hours, or we have to use 0.1 nitric acid HNO3. So either we can use that KNO3 0.5% solutions or 0.1 normal HNO3 solution. So this is mainly to overcome the dormancy related problems. The seeds are not given with these treatments. Mean if you go for sowing, you cannot get any seedling establishment. Even though the seeds are viable, they are not able to give rise any seedlings. So to overcome this particular dormancy, we have to give this particular treatment. And that next slide I will tell you some more treatments, and that treatment and all very useful for making your seed to perform very well under field conditions. And then now that the land is selected and the seed also ready, then we have to go for that transplanting. Before that, we have to look for the possibility of that isolations. Okay, we have to decide whether we are going to follow that space isolation or time isolation or barrier isolation. That and all we have to look for. If the facilities are available, so all the fields are owned by you, and there won't be any other varieties or in and around that your paddy seed production field, means better you can follow that space isolations. If at all any problem with reference to maintenance of that space isolations, in these cases you can follow that time isolation, particularly in the case of paddy. Okay, but officially seed certification department, they are permitting us to follow time isolation only in the case of maize, but in the case of paddy there is a possibilities are there, because this is also a self-pollinated one, and the flowering period also the 10 to 12 days. So because of that, no other ways are there. We can follow the 21 days of time isolations, or otherwise we can go for that barrier isolations. So for this purpose we have to raise that tall growing crops like sesbania, maize, sugarcane all around the four sides. So by that way, whatever that barrier crops are raised, so that will act as a trap crop, so that will prevent the entry of the foreign pollen into a seed field. So now we have to decide that isolation. So for paddy, it's a self-pollinated crops. There won't be much problems with reference to genetic purity maintenance. So because of that, for varietal seed production, both for foundation as well as certified class of seeds, we have to follow that 3-meter isolation distance. So 3-meter isolation distance is sufficient for paddy. 3-meter isolation distance is sufficient for varietal seed production, not for hybrid. Now we are discussing the matter related to that varietal seed production in paddy alone. Okay, so for paddy varietal seed production, we have to maintain both for foundation and certified class of seeds, we have to maintain 3-meter space isolations. And then these are all the matters related to that upgradation of the quality of the seeds. So before sowing, that seed must be given with some treatment. If possible, we can go for that upgradation of the seed quality. In the case of paddy, we are following that egg flotation technique. Egg flotation technique is followed for upgrading the quality of the seeds. In this, what we are really doing is we are preparing the salt solutions. So with the help of the salt solutions, we are going to separate the seeds fully packed with nutrients. Seeds fully packed with nutrients, and we are going to remove or reject the ill-filled seed or partly filled seeds or chappy seeds and all with the help of this one. So for this purpose, what we have to do is we have to prepare a salt solution with 1.13 specific gravity, 1.13 G. So in order to have a solution with 1.13 specific gravity, we have to dissolve 1.5 kg of edible salt, 1.5 kg of that salt, whatever salt is available in the house for cooking purpose, the same salt we can use. So we have to use 1.5 kg of salt that should be dissolved in 10 liters of water. So definitely that will give you 1.13 specific gravity. But here in the lab and all, we have that checking facility. Some instruments are available. With the help of that, we can check whether that particular water attain that specific gravity of 1.13. But at the farmer's point of view, they cannot purchase, all of the farmers, they cannot purchase that specific gravity, water specific gravity checking instruments. So for knowing purpose, what they can do is they can before addition of the salt in the water, so what they have to do is they have to take water in a bucket, 10 liters something. After taking up that good quality water in a bucket, they have to place a good quality egg in this water. If the water is good and the egg also good, means if you place the egg on the top, that will be settled at the bottom. Okay, that clearly indicates that water quality is good, the egg also good. Then after that you take out the egg and add the salt and go for dissolving. After addition and complete dissolving of the salt, you again place the egg in the water. So now that egg slowly started to take off and started to float. So that after first thing that may be here. Then after that you take out that one, add some more amount of salt and dissolve completely. Then again place the egg and check the position of the egg. Egg is remain here, means again you have to add some more quantity and go for dissolving. So whenever that egg that reaches that top layer and the new 1 rupee coin, the egg will be exposed to outside. So part of egg remain within the water and some part will be exposed to outside. The exposed area should be equal to a new 1 rupee coin. Okay, that egg will be float on the top. So at the time of flotation, that some surface of that egg will be exposed to outside the water. That exposed area should be equal to a new 1 rupee coin. So this is the position of the egg, means so definitely the water specific gravity will be 1.13 g. Okay, here that egg is used for knowing the specific gravity of the water after addition of that salt. Once this is the right position. So once this position is reached, once that this much level of that specific gravity of that water is arrived, we can remove the egg and place all your seeds, all your paddy seeds. So now only that good quality seeds alone will settle at the bottom. So the ill-filled one, insect attacked one, chappy one, everything will be float on the surface. So whatever that seeds that get floated, that should be removed. That will be used for some other purpose. Let's say example, we can use that one for making some parched rice. Tamil it is called as aval, or we can go for that cattle field preparation purpose, or we can use that material for that rice bran oil extraction, like that we can. So whatever that seed that settled on the bottom, that alone is a good quality seeds. So you have to separate that singers. After separation, that singers should be washed thoroughly, because here for separation purpose we are using salt solutions. So if you are not given with any washing mean, that salt will be there, that will create some sort of problem while you are sowing this particular seed in the field. Okay, so to avoid that particular problem, immediately after separation of that singers, so the whatever that seed settle at the bottom, that should be thoroughly washed to remove the salt. So this is a way in which we can upgrade the quality of the seeds. If you sow this kind of separated seeds, definitely that germination will be good, and whatever the seedlings that come out of that seeds, that will be a more vigorous, steady growth will be there. So this is a very popular technology, and most of the farmers also following this particular technology for upgrading the quality of the paddy seeds. And that is upgrading. Then again we are giving some treatment to make the seed to perform very well, so under field conditions. So some other micronutrient based treatment also we are offering. So mainly that 1% KCl treatment. So this is mainly for imparting that drought tolerance mechanisms. So to impart the drought tolerant mechanism and make the seed to germinate early under rain-fed conditions, before sowing, that seed must be soaked in 1% potassium chloride solution for about 10 hours. After that, we can dry the seeds under shade condition and bring back to the original moisture content. Then whenever that favorable condition arises, at the time we can go for that sowing. Or we can give that 2% monosulfur or 1% parasulfate solution based soaking. So this one also for about 10 hours. So this and all get infused and improve the performance of the seeds under field conditions. And then apart from nutrient treatments, some other protective treatments also we can offer. So that is mainly for giving protection for your seeds against soil-borne pathogen as well as soil-borne insects. So for normally for protecting the seeds against the soil-borne fungi, and in those cases we can give a carbendazim treatment. So here that recommendation is 2 gram per kg of seeds. Like that we can offer some fungicides and insecticide also we can treat. Similarly, we can infuse that biofertilizers also. So we can treat the seed with the azospirillum and astrobacter. The recommended quantity is 3 pockets per acre of seeds. So this and all biofortifications. So the microbes will be inoculated. The inoculated microbes will help the seed and the seedlings to fix the atmospheric nitrogen. So that will help the seeds and the seedlings. So these are all the advantages of that biofertilizer treatment. So after that, after giving all these treatments, here in the case of paddy, we are going for that nursery followed by the transplanting operations. Once you decided to follow that transplanting operation means we have to take up the nursery. So before sowing of the seeds in the nursery, and that should be given with appropriate soaking operations in order to make that seed to sprout. After sprouting only we have to sow the seeds in the nursery. So for making that seed to sprout, we have to soak the seeds overnight in a water, and that next day you have to take out the seeds along with initially that seed should be packed in a cunny bag. With that position we have to soak it in a water for about overnight. Then next day you have to take out that bag along with the seeds. That should be put it under dark. So during that times almost all the hydrolytic enzyme production and conversion process everything will take place, and once that conversion everything is over, that glucose will enter into the glycolysis process, started to produce that ATP, and the elongation of that embryo and rupturing of the seed coat and giving rise that plumule will occur. As and when that plumule just come out of that seed coat, at the time we can use that seed for sowing purpose in the nursery. So this is a free germinated seed. So in these particular conditions we have to sow the seeds in the nursery. So for paddy nursery we have to prepare a field that should be in a raised bed. We have to prepare that dimension of that raised bed and all 6 into 1.5 meter. The height of that bed also the width of that things also 0.5 meter wide things, and the channel also we have to arrange. So in this raised bed we have to sow the seeds. Normally for taking up cultivation of 1 hectare of land we need to have 50 to 60 number of beds with a dimension of 6 into 1.5 meters. So for each bed we can sow 500 to 600 gram of seeds. Each bed with a dimension of 6 into 1.5 meter we can accommodate 500 to 600 gram of seeds. So we are going to have 50 to 60 number of beds. Spare sowing is necessary. So per 1 cent 1 kg we can. Then some basal we have to apply DAP also at the rate of 2 kg per cent. Nursery we have to apply DAP. And 10 days prior to pulling we have to apply that gypsum also per kg per cent. So if we follow all these processes, that germination and seedling establishment will be there. Then as and when that seed attain optimum age we can go for that transplanting of the seedlings in the main field. In the case of short duration crops, that optimum age of the seedling is 18 to 22 days, whereas medium duration crop 25 to 30 days, whereas long duration the nursery period is 35 to 40 days. As and when that age of the seedling is 35 to 40, in the case of long durations we have to transplant the seedling in the main field. So before that seedling attain that particular age we have to prepare the main field. So we have to go for that puddling and plowing and everything. Finally we have to do that leveling, because most of the rice we are following that flood irrigations. So for in the case of flood irrigations, we have to ensure that leveled surfaces, generally that proper dispersal of water will be there, and there won't be any problem with reference to irrigating the field. So proper leveling we have to ensure, proper leveling, and also that should have adequate drainage facilities. So once that land is prepared, we can go for that pulling out of the seedlings from the nursery. So manually we have to pull out the seedling from the nursery, then we can go for that transplanting. So in the case of transplanting, we can follow that line sowing. Line sowing is recommended for seed purpose. We have to follow that line sowing, and spacing also you have to follow that optimum. Say example, if it is saturations, you have to follow that spacing of 20 into 10 centimeter. 20 means that is a spacing between the rows. 10 centimeter, that is a within the rows, that is a within the row between the plants. In the case of medium duration variety, we have to follow the 20 into 15 centimeter, whereas long duration, that is a 20 into 20 centimeter. Here that is guided. So 20 into 20 centimeter for long duration variety we have to follow. So line planting we have to follow. So here you can see that is a line planting they are doing. And then after planting, immediately we have to apply that all the major nutrients. So before that planting, that field should be applied with 12.5 tons per hectare. That is a farmyard manure. Farmyard manure we have to apply at the rate of 12.5 tons per hectare. And with reference to that macronutrient like NPK, nitrogen, phosphorus, and potash, as far as rice is concerned, the recommended dose is 150, 50, 50 kg of NPK per hectare. Out of 150, 50, 50 kg of NPK per hectare, 75 kg of nitrogen and 50 kg of phosphorus and 50 kg of potash applied as a basal. So entire dose of that phosphorus and entire dose of the potassium is applied as a basal. That is before that last bottling we have to apply that one. And the remaining quantity, remaining 75 kg of that nitrogen is applied in two split doses, one at tillering, second at flowering. So at the time of tillering we have to apply 40 kg, remaining 30 kg should be applied at flowering. The another thing is some of the micronutrient also, micronutrient deficiency also creates some sort of problem, particularly in the case of paddy zinc deficiency, that causes problem of the T.Eevera disease. T.Eevera disease, actually disease, it's not a disease, but it's a nutrient deficiency related symptoms that will affect the yield potential of the paddy. In order to overcome that particular problems, as a basal, that is just before that last falling, that field should be applied with 25 kg per hectare zinc sulfate. So per hectare we have to apply 25 kg zinc sulfate as a basal. So by that way we can overcome that particular problem. Or once you miss to apply that zinc sulfate as a basal, you can apply that one during 30 or 40 and 50th day after planting as a foliar spray. So as a foliar spray you can apply 30th day, 40th day and 50th day after planting. The concentration is 0.5%. So you have to use 0.5% as a foliar spray. So mainly to overcome the zinc deficiency related things. So these are all the macro and micronutrient requirement as far as the rice is concerned. And then coming back to that crop management activity, so that weed management is very very important. So here to control that weed, we have to apply the pendimethalin at the rate of 2.5 liters per hectare. Similarly that putachlor also, this and all some free emergence herbicides. So putachlor 2.5 liter per hectare. So this will control that weed growth. And then on 30 to 34th day after planting, manually we have to do that one hand weeding also. If we follow, we can automatically control that weed population. So this is a very very important one. Then irrigation also another very very important operation, because we are cultivating these particular crop for seed purpose. So we have to provide adequate amount of water during the critical period. So critical period we have to ensure that water availability. If you are not able to ensure availability of water during that critical period like primordial initiation, vegetative and milky and maturation stage, ultimately that quality of the seeds get affected. Primordial initiation means that is a transition period from vegetative to reproductive. So by the way of dissecting that stem of the plants, we will come to know whether this is getting transformed from shifting from vegetative to flowering and all. Okay, so during that particular time we have to ensure that adequate water supply, and vegetative then milky stage and maturation stage and all very critical stages. Here you can see that various stages of the development of the paddy is given, so that panicle formation, flowering, maturity, everything is there. So during that critical period we have to ensure that water availability. And that previous that agronomic principles related to that seed production itself we discussed that rogging is very important operation, water maybe the crop. So in the case of paddy also we have to get into the field, particularly at the time of flowering and maturity times we have to get into the field, and we have to verify the presence of the field and percentage of op types that is available in the field. If at all any op types are available, that should be removed. So by that way only we can ensure that the genetic purity of our seed material. Rogging is very important operations. Likewise that pest management also very important operations. So we have to use appropriate chemical to control the major pests related to that paddy, like stem borer, thrips, leaf hopper, all those things we have to control. So appropriate pest control measure also we have to follow, and these are all the major pests with reference to that paddy is concerned: PPH, leaf folder. So all those things we have to control. And then similarly the disease also, blast, brown spot, sheet blight, bacterial leaf blight, like that. So many pathogen also causes problems, so we have to control all these things, or otherwise the physiological quality of our seed get affected. Physiological and physical quality of our seed will be affected. If it is a seed crops, so that should be subjected to that field inspection. In the case of paddy, that seed certification officers, they will visit your field during flowering and maturity time. So during flowering and maturity time they will come for the inspection. So they will get into the field and they will point out that field standard, mainly that percentage of op type and objectionable weed plants, all those things they will verify. In the case of foundations, the maximum permissible limit of that op type is 0.05%. The maximum permissible limit, up to this they can permit 0.05% is the maximum permissible limit. Suppose that op type number exceed beyond that 0.05%, in the case of foundation seed or paddy, means simply they will reject your field, or during that first inspection they will ask you to remove all those things, then only I will accept. Like that they will give you a warning, because it is a self-pollinated crops. If at all any op type is there, that going to be physically there. So because of that they give a warning. If it is a cross-pollinated crop, if at all any op type is recorded after that pollination and flowering is over, definitely they will reject the field. Since it's a self-pollinated crop, because of that they will give a warning. So if it is a foundation, the maximum permissible op type is 0.05%. If it is a certified seeds, there they are permitting up to 0.2%. Similarly that objectionable weeds also they have limitations. If it is a foundation, maximum permissible limit is 0.01%, whereas certified seeds 0.02% is a maximum permissible limit. So during that flowering time and maturity times they will verify and they will compare your field standard with the standard given in the Indian minimum seed certification standard. So for all the crops, the field standard and the seed standards and all available in the name of Indian minimum seed certification standard. So whatever that standard is available, that is a standard. So your field data will be compared with the standard. If it is your field data is on par with that standard, then only they permit you to continue that seed production operation, or otherwise they will reject your field and ask you to convert your seed production field into a grain production field. So maintenance of that field standard is very important. Then that everything is okay, that field standard everything is fulfilled, both at flowering and maturity time, that seed certification officers they will permit you to go for that harvesting. So you have to, since it's a seed crop, we have to harvest the crop at a physiological maturity stage. So that symptoms associated with the physiological maturities are the seeds turn straw yellow color, straw yellow color, and also the ear gate exhibit that drooping symptom, that fever that will be erect or something. So as and when that attained physiological maturity, that ear gate started drooping symptoms, and moisture content also somewhere around 18 to 20%. So these are all the maturity indices with reference to that paddy attained physiological maturity. So during that times we have to harvest the crop and bring down that moisture content, then go for that separation of the seed from the mother plant. Once the seed is separated from the mother plant, then you have to reduce that moisture content to the level of 10 to 13%. Then you can go for that cleaning and grading operations with the help of the machines. So in the case of paddy, we have different material like long slender material, medium slender, short pole, like that. So in Tamil Nadu, that long slender is the most preferred rice for consumption purpose. The very good example is ponni and improved white ponni. So long slender means the dimension is 1.3 millimeter into 19 millimeter. Slender type, and medium slender, short pole. Short pole is normally most prepared food item, most prepared rice by the Kerala. So it's normally in Tamil it is called as a gundu rice, short pole type. Seed yield will be somewhere around 50 to 60 quintals per hectare. So that is a 5,000 to 6,000 kg per hectare in the case of variety. And once that cleaning, grading, everything is over, after that we have to go for that storage. So before storage of your material, that should be given with some protection treatment, because during storage there is a chance for occurrence of the pest and diseases. So in order to give protection to your seeds against storage pathogens as well as that insect populations, seed must be given with pesticide as well as fungicide treatment. So fungicide normally used for these purposes pevistine. So previously we recommended Thiram. Recently Thiram and Captain and all burned by the government of India. So now better we can use that carbendazim or pevistine at the rate of 2 gram per kg of seeds. Then after treatment is given, we can pack the seeds in appropriate containers. Say example, that duration of that storage is known. Accordingly we can select that that containers used for storage. Say example, you are going to store the seeds for about 6 months. Short term means in that case you can use that moisture pervious container. Moisture pervious container means this container will permit the entry of the moisture into the inner side of the container. Say example is the candy bag or cloth bag. So here you cannot maintain the moisture content of the seed throughout the period of storage. So because of that, this particular container is used for short term storage. Medium term storage means there we have to use that polythene bag with less than 300 gauge thickness. So that is a thing. Long term storage we have to use that moisture vapor proof container. So here that entry of water both in the form of liquid as well as vapor is prevented. So whatever that initial moisture, say example the initial moisture content of the seed is 6%, means even after a year of storage that moisture content remains same. That is the advantage of this one. So if you are aiming for storage of seeds for more than a year, you can use that moisture vapor proof container. So the example is aluminum foil pouches or polythene bag with more than 300 gauge thickness. So these are all the things. So based on the duration of storage, you have to select that appropriate storage containers for packing and storing up your seeds. So before selling up your seeds in the open market, that should be tested with reference to their seed quality parameters. So the seed standards need to be verified. The seed standard like physical purity, genetic purity, and germinations, moisture content, all those things need to be verified. So as far as that paddy is concerned, for foundation seed, purity wise, that should have 98% both for certified and foundations. And the maximum permissible inert matter other than your seeds, the maximum permissible limit is 2%. And huskless seeds in both the cases, foundation certified classes 2%. And other crop seeds in terms of number, maximum permissible limit is 10 number per kg in the case of foundation, whereas in the case of certified we are permitting up to 20 number per kg. Then other distinguishable variety, in short call it as a ODV. It is also maximum in terms of number. In the case of foundation seed, the maximum number, permissible number is 10 number per kg. Certified seed 20 number per kg. And above all, whatever may be the class of seed, either it's a brooder seed or foundation or certified, that should have a 80% germinations. There won't be any difference among the classes with reference to germination. So that should be minimum 80% germinations. Then only you can sell that particular seed as a seed in the market. Suppose the moisture and the germination is less than 80%, you cannot sell that particular seed in the market. Similarly, that moisture content of the seed also we have to ensure. So both for foundation certified seeds, if you are going to use that moisture previous container, that is a candy bag, means there we have to ensure that 13% moisture content. If it is a vapor proof container, the moisture content should be 8%. So these are all the things with reference to seed standard. So here at the field level, we have to maintain that field standard. Once after harvesting and threshing, cleaning and grading, everything is over, before packing and storage, and seed quality need to be tested. That seed standards we have to check. So whatever that test results you are going to get for your seed, that should be compared with the Indian minimum seed certification standard. So these are all the standards. So this you have to compare the data of your seed lot with this standard. So if your seed lot fulfill all these requirements, then only you can sell this particular material as a seed in the open market. So these are all the items with reference to that paddy varietal seed production is concerned. So in the next class we can discuss about paddy hybrid seed production. Thank you.